All right, it's 8 o'clock, so we'll get started. Thank you very much for attending in the morning session here. So we're really excited to present this session to you, Abstract Session 1. This has a rich diversity of talks ranging from pancreas cancer to detection and basic and translational science all the way to clinical medicine. And so my name is Josh Datta. I am a pancreatic and hepatobiliary surgical oncologist at the University of Miami. I also run a lab that studies the intersection of myeloid immunobiology with the tumor microenvironment. And I'm thrilled to co-moderate this session with Dr. Karina Yoon, who will be introducing herself in a couple of minutes. She's from the University of Alabama at Birmingham. So a couple of ground rules before we start. This is a packed session. We have 12 speakers to cover in 2 hours. So we have 10 minutes per talk, 7 minutes for your talk and 3 minutes for Q&A. And so our charge is to make sure that you stay on time. So if we are a little militant, we apologize in advance. So please be respectful of your other speakers and our audience members, and please try to stay on time. To expedite things, I will actually introduce the title of your talk as well as your name and your institution, so you can dive right in. All right, so without any further ado, we'll start with our first talk that is also presented by a young investigator. It's titled Combined MEK and STAT3 Inhibition Reprogrammed Stromal Inflammation to Overcome Immunotherapy Resistance in Pancreatic Cancer by Dr. Vanessa Garrido. This talk literally hits close to home. She's from the University of Miami.
So good morning, everyone. I'd like to thank the opportunity to share my work here today. So as we know, pancreatic cancer is very resistant to chemotherapy. And one of the major contributors to this therapeutic resistance is activating KRAS mutations that actually drive malignant progression and have been shown refractory to target therapy. So recently, a lot of attention has been focused on CAF heterogeneity and its role in immunotherapy resistance. So several studies have shown that in the tumor microenvironment, there are different subsets of cancer-associated fibroblasts, including pro-inflammatory CAFs, also known as ICAF, LRC15 positive myofibroblastic CAF that recently have been correlated with poor response to immunotherapy, and some studies have shown the dissipation of putative tumor-restraining CAF subtypes during tumor progression. Beside that, the profoundly immunosuppressive tumor microenvironment is characterized by high prevalence of myeloid-derived suppressor cells that can impair T-cell infiltration and activation. So to successfully treat pancreatic cancer, we need therapeutic interventions can actually overcome all these three factors. So our lab has long standing efforts in targeting oncogenic RAS signaling in pancreatic cancer. And in this paper published in 2018, we showed the inverse correlation of STAT3 and MEK signaling mediating resistance to RAS inhibition. So we showed that when we combine MEK inhibitors and STAT3 inhibitors, we have like a huge decrease in the tumor burden. In these studies, we also observe a profound effect of MEK-STAT inhibition on remodeling the stromal fibrosis, here shown by the decrease of trichome blue, alpha-SMA, and serored staining. So, give these results, we ask, can MEK-STAT3 inhibition unmask CAF heterogeneity to improve immunotherapy sensitivity in pancreatic cancer? So to address this question, we perform single-cell RNA sequencing in PKT mice treated with vehicle or MEK and STAT3 inhibitors. And when evaluating the CAF compartment, we actually observe like a very distinct transcriptome between these two treatment groups. So the deeper analysis of the CAFs actually reveal extreme downregulation of ICAF-defining genes like CXCL1 and IL-6. So these findings were then validated by qPCR in whole tumor lysates, which confirmed those results. So then we asked if MEK-STAT inhibition could be acting in ICAF polarization in a CAF-dependent manner. So to interrogate that, we performed CRISPR-Cas9 gene editing of MAC1 and STAT3 in calf derived from KPC tumors. We later co-injected these knockout calves with tumor cells in black 6 mice. So using flow cytometry, we investigated the presence of 3 previously described calf subtypes, and we found a decrease in the inflammatory calf, as well ICAF to MyCAF ratios in these tumors in the presence of calf knockout MAC and STAT3. So to evaluate if MEK and STAT inhibition can shape calf heterogeneity beyond ICAF polarization, we actually did the subcluster of the calf compartment and found 4 different calf subtypes. So using their differentially transcript signature, we named those calf clusters, and CAF4 was named myofibroblastic calf. So interestingly, LRC15 expression, which I told you before that is related to immunotherapy resistance, it was enriched exclusively in this CAF4 cluster. So CAF3, we named it antigen-presenting cells. The CAF2 were secretory and inflammatory calf. And CAF1, which was enriched by CD34 expression and Ly6A, both are markers for stem cells. We named this calf mesenchymal stem-like cells. So some studies have shown actually that this mesenchymal stem cell-like calf, they have some tumor restraining functions and have been correlated with like facilitating the therapy sensitivity in cancer. So later we did pseudotime lineage reconstruction and actually found that these mesenchymal stem cell-like calf are the origin of all other calf subtypes. So having this minor bifurcation in the secretory inflammatory calf and another one in the myofibroblastic calf. So when we evaluated the difference in the calf cluster between the treatments, we observed that in vehicle-treated tumors, we found this enrichment for myofibroblast calves, secretory inflammatory and antigen presenting. And in MEK-STAT treatment, actually, there's an ablation of these 3 types of calf and an enrichment for these mesenchymal stem cell-like calves. So taken together, our data suggests that MEK and STAT3 inhibition actually attenuates this pro-inflammatory calf, reduce LRC15 myofibroblasts, and while enrich for this putative tumor restraining mesenchymal stem cell-like calf subtype. So having this effect on the stroma inflammation pancreatic cancer, we evaluated the CD45 cells in the tumor microenvironment, the immune cells, and we found that with MEK and STAT3 inhibition, there's like a huge decrease in the myeloid compartment, here shown by F480 staining, CD206, the M2-like macrophages, and also MDSCs. At the same time, we saw a big increase in the T cell populations, both CD4s and CD8s, as well as some activation in this in some T cell populations. So after showing that MEK and STAT inhibition can potentially overcome these 3 major contributors of CAF therapy resistant in pancreatic cancer, we hypothesized that if we add MEK and STAT treatment with anti-PD-1 therapy, it could increase sensitivity of pancreatic cancer to checkpoint inhibitor and immunotherapy. So when we treated PKT mice with the triple combination, we saw that while PD-1 and vehicle, there was no effect on the tumor growth, MEK and STAT inhibition, there's like a reduction of the tumor area, about like 15 and 20%. But the triple combination, there was like a even further reduction in tumor area. And more importantly, we also see that the triple combination has like improved significantly the survival of PKT mice, even compared to MEK and STAT and other 2 treatments. So we are also able to evaluate the effects of this triple therapy in a patient that was had chemotherapy refractory disease in metastatic pancreas. And after 3 months of treatment, we actually observe a profound reduction of the tumor size, like nearly 50% of the tumor was and then also a radiological response and metabolic response. And there was no adverse event in this patient, which was good. So in summary, I have shown here today that combined MEK and STAT inhibition uncovers thromboplasticity by attenuating ICAF and LRC15 ICAF phenotypes while enriching for this mesenchymal stem cell ICAF. It also attenuates immunosuppressive MDSCs and tumor-associated macrophages and enhance the trafficking and function of activity of CD8 T cells. So we also showed that MEK and STAT inhibition in combination with PD-1 block can overcome immune checkpoint resistant in pancreatic cancer. And we are also able to show the clinical efficacy of this triple combination in patients. So I'm proud to announce that actually our data has been recently published in Gastro this month. And then you can check out the full manuscript using the QR code here. I would like to thank my mentor, Dr. Nipul Merchant, the other PIs in the lab, Dr. Datta and Dr. Nagatihali, also the other lab members. And thank you.
Great. This paper is open to the floor. If there are any questions, please come up to the microphone. While we're waiting for people to come up, how do you reconcile this putative tumor restraining CAF population with this concept of the universal CAF that's been recently proposed by the Turley group? And how do you think that plays into therapeutic resistance?
So actually, that's a good question. We actually did an investigation in those CAF compartments, and actually they have like several similarities, but we also see a very distinct transcriptome between the two. So we are now evaluating this further in our lab, doing some mouse models, and then we would be able to answer that question.
Outstanding. Oh, there's a question.
Just one more. Hi, very interesting work. Just wonder if you saw the personal fibronectin. I didn't see listed in one of those subclass of fibroblasts that you mentioned. Did you mention fibronectin?
Sorry, could you speak into the microphone? We couldn't hear you.
Yeah, very nice job.
Thank you.
I just wonder if you saw an increase in fibronectin, the personal fibronectin, one of those subclass of fibroblasts that you found.
So actually, we didn't check for that, but we are doing like a deeper investigation of the whole fibrosis in the tumor, and then I think we're going to have that question later.
Yeah, because I saw an increase in fibronectin and it surprised me that you don't measure that. Thank you.
Thank you.
Thank you again. We're going to move on to our second talk. This is also by a young investigator, Dr. Zhu from the University Clinic in Heidelberg. She is going to be presenting on persister cell phenotypes in pancreatic cancer and how they contribute to poor patient outcomes after neoadjuvant therapy.
Oh, sorry. Good morning, everyone. It is my great honor to attend the APA meeting and to present my study here. Pancreatic cancer is a malignant disease with increasing incidence and almost paralleled mortality. It has now become the #5 cause of death worldwide. Unfortunately, only 20% of the patients are suitable for resection surgery, followed by adjuvant chemotherapy. The rest are all non-resectable cases and can only receive neoadjuvant chemotherapy. Both of the treatments can significantly prolong patient outcomes. However, the chemotherapy has its limitations due to a wide variation of tumor heterogeneity and patient different response to drugs. So take one step further, the molecular subtyping derived from the transcriptomic analysis may enable us to have translational insight into the pancreatic cancer. There are several molecular classifications with prognostic potentials. In general, classical molecular subtype is correlated with a longer survival, while basal molecular subtype is correlated with a shorter survival. However, all of these classifications were established in patients after adjuvant chemotherapy. So our question is, does this also apply after chemotherapy in the neoadjuvant setting? So previous studies have shown that chemotherapy may enrich certain different cell phenotypes, which were called persistent cells. The question is, it is uncertain whether these persistent cells also arise in PDAC post-chemotherapy, and if so, what is the functional significance? So our aims are to determine whether molecular subtypes are altered after chemotherapy, and whether persistent cells become enriched after chemotherapy, and if so, what is their physiological functions? To solve these problems, we gather two different types of patient samples, chemo-naive and post-chemotherapy. For the post-chemotherapy group, we further separate them into M4-FOLFIRINOX treated group and the gemcitabine treated group. We did the RNA sequencing and find that chemotherapy alters neoplastic and stroma cell gene expression programs compared to chemo-naive tumors. We also applied our data to morphic subtypes and find that the basal molecular subtypes were enriched in the post-chemotherapy samples. And morphic subtypes can prognosticate the patient's outcome in neoadjuvant but not post-chemotherapy cohorts. We then detected the protein expression of some potential markers by multiplexed immunofluorescence. We found two surrogate markers, Datta6 as a classical marker and keratin 17 as a basal marker. We found that Datta6 and keratin 17 positive cell populations persisted after neoadjuvant chemotherapy. We also identified another marker, CYP3A, which is co-expressed in Datta6 and keratin 17 positive persistent cells. These cells were also enriched in the post-chemotherapy samples, especially M4-FOLFIRINOX treated group. CYP3A is also associated with reduced survival in patients preoperatively treated with M4-FOLFIRINOX, but not gemcitabine. CYP3A is actually a metabolizer associated with irinotecan resistance. It can turn irinotecan into non-active metabolites. So we applied the tandem mass spectrometry on the human-derived organoids. We found that CYP3A highly expressed organoids were more resistant to irinotecan. So according to the results, we get the conclusion that after neoadjuvant chemotherapy, basal-like subtypes were enriched. Datta6 and keratin 17 positive persistent cells, which also co-highly express CYP3A, were enriched after patients treated with M4-FOLFIRINOX. Classical molecular subtypes predict the patient outcomes after adjuvant but not neoadjuvant chemotherapy. Neoadjuvant treatment reprogrammed neoplastic and stroma cell gene programs. Functional organoid experiments demonstrated that CYP3A high-expressing cells were associated with resistance to irinotecan and associated with poor clinical outcomes. Datta6, keratin 17, and CYP3A are markers of prognosis and M4-FOLFIRINOX drug response and may contribute to clinical treatment selection. So further study of persistent cells may result in a better methods of patient treatment with pancreatic cancer. Thank you very much.
This paper is open to the floor. While we're waiting for folks to come up, do you have any paired samples from the same patient, match patients pre and post treatment? to understand whether this biology is similarly happening in these patients?
Yes, this is a retrospective study, so we could only collect from different samples, chemo-naive and post-chemotherapy. But it is ongoing. We're trying to get some biopsies of patients before chemotherapy and then compare after resection. So this is ongoing. We will see.
And since we have a couple more minutes, we want to give you your 10 minutes. Any other questions from the floor?
I have a question. So you're talking about CYP3A. It's
Yes.
a cytochrome P450 related enzyme.
Sorry?
CYP3A
Yes,
is a cytochrome P450
Yes,
related enzyme. So
exact-
is this something to do with the drug metabolism then? Overexpression of this particular enzyme, CYP3A, and irinotecan expression?
Yes, it actually is a metabolism associated with irinotecan resistance. It will turn irinotecan into the non-active metabolites like APC, NPC, M4 instead of the active molecule SN38. Yeah, thank you.
So, you know, the Boston group from Bill Huang and others actually had a very similar paper that they recently published in Nature Genetics, where they saw enrichment of neural-like progenitor-like cells
Mhm.
after, you know, treatment trajectories with chemo and chemoradiation. I wonder if in these persister populations, you see any neural-like phenotypes. Have you looked into that? Perhaps there's some similarities in the biology.
Well, I think those markers are the what we found for the persistent cells, but we also find they are with high phenotypical plasticity. We can see by the immunofluorescence, if that's what you're
In
asking.
terms of their transcriptome, do they enrich for neural-like markers?
I think
Because you've done transcriptomic studies as well, correct?
yes,
Yeah.
yes. I think so, but we didn't typically pick up those cells, but according to our transcriptomic data, yes, those markers are also enriched.
So,
We have a question from the floor.
so first, thank you for interesting talk and a lucid presentation. Did you have a chance to look at the immune cells and the cancer-associated fibroblast subtypes? Because they've been known to alter with treatment also.
Thank you very much. This is exactly the next step that I would like to do to detect the immune cells of fibroblast and tumor microenvironments surround these persistent cells. Thank you.
Congratulations.
Thank you.
Okay, so my name is Karina Yoon from the University of Alabama at Birmingham. I'm glad to be here to introduce the next speaker, Dr. Jingshan Yang from the University of Oklahoma Health Science Center. The title of his talk will be "The Circular RNA NFC7 Inhibits Tumor Growth and Ameliorates Pancreatic Cancer Cachexia via PHLP-PS-AKT TGF-beta signaling axis." Dr. Yang?
Good morning, everyone. I would thank APA for give an opportunity to present our recent study on the circular RNA in pancreatic cancer. So there's some background information. So circular RNA belong to a big long non-coding RNA family, and they are single-stranded covalent closed RNA molecules with no open end. And one of the major circular RNA function is to act as a sponge to the microRNAs to influence the target genes expression. Our group, Dr. Min Li's lab, so we have a long time interest in a zinc transporter called ZIP4. So it's a zinc transporter. We found ZIP4 plays an important role in promoting pancreatic cancer progression, and also it can activate the miR-373 pathway to regulate the pancreatic cancer apoptosis procedure. Recently, we also found that ZIP4 contribute to the pancreatic cancer-induced muscle wasting, which called cancer cachexia. So this project I'm going to present is just to investigate if any circular RNA could just play a role in this ZIP4 induced promoting pancreatic cancer progressions. So first, we screened top 100 circular RNA differentially expressed in 6 paired human pancreatic cancer tissue and surrounding normal tissue. And we found 5 circular microRNA which contain the potential microRNA 3 binding sites. Well, only one of these circular RNA is called synapse circle synapse C7. When we just do the microRNA pull down assay, we found that this synapse C7 was significantly enriched in the microRNA 373 captured fractions. Further, we do the RNA immunoprecipitation with AGO2 protein, and we found that this synapse C7 could significantly increase upon the MIR373 mimics in the pancreatic cancer cells. The luciferase assay also confirmed this wild type synapse C7 microRNA can be significantly inhibit the luciferase activity in microRNA 373. When we overexpression this circular RNA in pancreatic cancer cells, we found that it can significantly inhibit the cancer cell progression and also the colon formation. When we analyze this human tumor suppressor genes, we found there are only 3 possible microRNA 373 target genes was negatively regulated with ZIP4 expression, and this PHRPP2 was the only one which can show significant induction upon the circular NAP67 expression. The microRNA pull down assay confirmed this PHRPP2 mRNA can directly bind into the microRNA 373. So when we overexpression this circular microRNA 373 in tumor cells, and the xenograft tumor shows that increased Ki67 accompanied with the lower expression of the PHRPP2. The similar thing we also found in the human PDAC tumor tissues. We found the PHRPP2 level was negatively correlated with the ZIP4 expression. And when we overexpression the NAPC7 circular RNA in cancer cells, it increased the PHRP2 expression and also significantly inhibit the tumor growth. Interesting, we also found in those circular RNA overexpression tumor mice, they have increased the body weight, and also their skeletal muscle, the TA muscle and the GS muscle also show increased muscle mass compared to the control group. When we analyze the muscle tissue collected from those mice, the overexpression circular RNA group shows increased myosin heavy chain protein level, and also it reduced the estrogen-1 protein level on Western blot, which is an indication of the reduced proteolysis in the muscle cells. And we also find that in tumor tissue, it shows significant less TGF-beta expression in the circular RNA overexpression tumor tissue compared to the empty vector group. So our working hypothesis of this finding that this circular RNA is the circle NAPC7 could inhibit the ZIP4 induced pancreatic cancer tumor growth through binding to microRNA 373. Then it will lead to increased expression of the PHRPP2, and PHRPP2 is a known AKT phosphatase. So it further just leading to reduce the phosphorylation of the AKT and cyclin D1, leading to inhibition of the tumor cells proliferation. In the meantime, this circular RNA could also inhibit the ZIP4 induced muscle wasting through inhibition the PHRPP2, then phosphostatin 5, resulting decrease the level of the TGF beta in the in the mouse and leading to reduce the muscle wasting in the mouse model. And here I just want to thank my mentor, Dr. Min Lee, and also all our lab members. And what I want to do, especially is point out that I want to make a big shout out for one of our postdoc, Dr. Xiuhui Shi. So she is the one perform the majority of the work, but she want to present her new project, I think, about ZIP4 promoting GDF15 secretion by the tumor-associated macrophage in pancreatic cancer cachexia. So she will present her abstract, I think, for tomorrow. It will be I think 178. So hopefully everybody can just go there to take a look. Thank you.
Thank you, Dr. Yang. Any questions from the audience, please? One question from.
It's Fred Gorlick from New Haven. It's a very, very interesting observation and work. And I was wondering, I missed it, is the circular mRNA made endogenously? And that's number one. Number two, can you engineer these basically to include an inhibitory domain? In other words, generate an artificial circular mRNA and deliver that to a system?
Yes, this circular RNA actually is a natural product. So it's first, I think when we discovered, people believe that it's like other long non-coding RNA. They think they don't have any functions. But I think now, recent years, I think there's quite a few groups. I think they did the detailed mechanism study. They found that this one was the circular RNA was it's an alternate splicing product through the normal mRNA transcription. Then they just do the splicing with the different proteins, so they can just form this circular one. And yeah, I think there's a group. I think that I know. I think there are two major groups. I think they were starting to do this kind of the therapeutic application of the circular RNA. So they do modify the sequence of the circular RNA. So they try to just target certain mRNA to do some anti-cancer treatment.
Thank you. Next question.
Hi, Jason Pederesi from UMass Medical School. So really excited to hear some very cool cachexia stories. I'm going to talk tomorrow about an adipose tissue wasting phenotype with PTHRP, which is also a TGF-beta miR-373 driven gene. So I was wondering, do you see only skeletal muscle wasting, or do you also see fat wasting or fat browning in any of your phenotypes?
Yeah, that's a great question. Actually, yeah, that's what we were just try to look into the adipose tissue wasting. And also, I think from our previous study, I think not only focus on that, but when we do the cachexia study, we found that the mice, especially those xenograft ectopic or xenograft tumor mice model, we can see clearly, I think, early stage, let's say maybe 10 to 2 weeks, 10 days to 2 weeks, we can already see significant adipose tissue wasting first. And then usually, I think for the muscle wasting part, skeleton part muscle, we usually observe those kind of muscle wasting syndrome, I think around 3 to 5 weeks. So it's a late stage of the cancer development.
Great talk.
Thank you.
Thank you.
Thank you, Dr. Yang.
All right, moving on to our next talk. This is going to be a detection talk in the detection space by Dr. Bergamashi. I'm sorry if I butchered your name, from Blue Star Genomics, and she's going to be presenting on validation of a blood-based early detection pancreatic cancer test in a high-risk population.
Good morning, everyone. I'm going to switch topic compared to what was presented earlier on, and I will be focusing on early pancreatic cancer detection. And really, I wanted to start remarking why it is so important to actually focus on early cancer detection. Only 11% of pancreatic cancer are actually detected in a localized stage. More than 50% are detected when the disease is already distant. And we know that there is a very poor 5-year survival equal to 3%. Current methods are actually not sufficient to detect early cancer, either for lack of ease for broad implementation or for lack of specificity. Here at Blue Star, we actually focus on early detection of pancreatic cancer, and we're actually leveraging a novel technology that uses this epigenomic signal that can be detected in a blood-based manner. Briefly, I'm going to walk you through our technology. Our liquid biopsy test, again, is based on the capability of detecting cell-free DNA fragments that are shed by tumor DNA and being able to detect this epigenomic signal. This epigenomic signal is called 5-hydroxymethylcytosine, and I know it's a mouthful, so we are shortening down to 5-HMC. 5-HMC is the product of oxidation of the 5-methylcytosine that is actually derived during the process of demethylation. And this process is carried out by the TET enzyme. And it's really important because it allows to switch from a state of gene suppression to gene activation. And indeed, the 5-HMC marks those genes that are either activated or poised for activation. And by detecting these 5-HMC marks, you are actually detecting active biology differently from other methylation methodology that are out there. So now I'm also going to walk you through how we develop our pancreatic test. And we started with a training set of 132 pancreatic cancer and 528 non-cancer. From each individuals, we receive blood that was processed in our CLIA-CAP laboratory in San Diego. And then from the blood, we actually process to plasma to then enable the isolation of cell-free DNA. About 10 nanograms of cell-free DNA enter the process and is sufficient to generate whole genome sequencing libraries. From the whole genome sequencing libraries, then we apply a click chemistry that enables the specific and selective enrichment of the 5-HMC signal. Then both WGS and 5-HMC library are sequenced, and then after sequencing, the data is pushed through our machine learning modules that really enables to detect and selectively pick out those features that best can differentiate between pancreatic cancer and non-cancer. Those features then they represent the so-called classification algorithm. And really the classification algorithm readout is a prediction score. And based on this prediction score, we can define what is classified cancer versus non-cancer by setting a cutoff. And this cutoff is actually a threshold that was represented by the 98% specificity set on the training set of the non-cancer samples in our cohort. Now that we have a lockdown laboratory process and we have a lockdown algorithm, we went to validate our performance in a large case control study. The validation data set included more than 2,000 patients of about 2,048 non-cancer and about 102 pancreatic cancer. The other thing that I wanted to point out is that within this validation set, we included patients with different risk for pancreatic cancer, starting with family history, genetic predisposition, new onset type 2 diabetes, long-standing type 2 diabetes, heavy smoker, but also individuals for which there is no known risk associated with pancreatic cancer. On the right-hand side, you can see the validation performance, and the early stage sensitivity that we measured was equal to 68.3%. And the specificity of 96.9%. We also wanted to compare the performance of our pancreatic cancer test with a very well-known CA19-9 biomarker. And we did so by selecting a subset of the validation cohort for which we actually ran CA19-9, and we had obviously a performance using our 5-HMC based test. The study cohort comprised of 580 patients, 503 non-cancer and 73 pancreatic cancer. it was really exciting to see that actually our pancreatic cancer test was outperforming CA19-9, especially in the early stage sensitivity. In fact, we saw an increase, an improvement over CA19-9 of about 31.6%. Now, the big question is, how do we implement an early cancer detection test? Obviously, the individuals that most will benefit from an early detection test will be those that are at highest risk for the disease. In this slide, I am actually representing different pancreatic cancer risk, and on the y-axis reporting out the level of risk, and then it's color-coded by the number of individuals within each bucket. We heard yesterday at length about the risk for chronic pancreatitis, genetic predisposition, and we also heard about the risk of pancreatic cancer in those individuals that have been newly diagnosed with type 2 diabetes. This is actually a very interesting population. About 25% of all pancreatic cancer actually have been diagnosed within 36 months of a type 2 diabetes diagnosis. And also very interestingly, these individuals, they actually, within this population, about 1% will develop pancreatic cancer in a 3-year span. So we are actually focusing in this population because as you can see, about 1 million every year will be diagnosed with type 2 diabetes, and having a 1% incidence of pancreatic cancer really provide a great opportunity for early detection. So we have designed and launched this clinical validation study for this population. It's called NODMED, New Onset Diabetes Management for Early Detection of Pancreatic Cancer. Is currently recruiting, and we are planning to accrue up to 6,000 patients. We are planning that every enrolled patient will be undergoing multiple blood draws, and in case of our pancreatic test coming out as positive, they will be sent for an MRI. The study objective is actually to evaluate the clinical performance in terms of specificity, PPV, and MPV, and then also evaluate stage shift. Now, in conclusion, I've shown you that we have developed and validated an early detection pancreatic cancer test that is based on the analysis of a 5-HMC. We can do that with a simple blood draw, and we have shown high specificity and early stage sensitivity. We also show that our test is superior to CA19-9 in terms of early stage sensitivity. The test is currently undergoing clinical validation in the NOD population, but we are also exploring to expand the testing and the validation in other high-risk population. I also wanted to mention that the test is currently available for early access, and for any more information, please refer to the website that is referred to the slide. Thank you.
This paper is open to the floor. I think we have a question. Please introduce yourself when you ask your question so that everyone can know who you are. Thank you.
Sure. Shaunak Majumdar from Mayo Clinic. Nice presentation. Thanks for sharing your data. My question is, you call it a 5-HMC based test. Can you just expand on what exactly is being measured? That's number one. Number two is, what was the AUC for CA19-9 standalone in your experiment?
Okay, so in terms of what what we're measuring in terms of the 5-HMC, so as I mentioned, there is this modification on the metal cytosine, and we are able to capture those fragments of cell-free DNA that have this modification on this cytosine, right? So we are picking it out and we are sequencing it, and then we are comparing to the non-cancer. So we do a genome profile of this all 5-HMC throughout the genome, right? And then we are comparing to the non-cancer and really selecting only those 5-HMC genes or genomic features that specifically differentiate pancreatic cancer versus non-cancer. Because we have seen, for example, from tissue data that there is a specific profile that differentiate pancreatic cancer, for example, from normal tissues from pancreas.
Yeah, and the question on what was the AUC of CA19-9?
That's a great question. I do not remember that. I need to go back and check on that one.
Thanks.
We have time for one more question
This
in
will
the
be
interest of time.
A brief question, Suhail Hussain from Stanford. Thank you. It's a real technical question about the workflow sample processing. For this sophisticated assay, what is the time frame for storage of the sample after venipuncture or processing? Because that may be a practical issue.
Yeah, so we are collecting the samples in Strack, so that will maintain the sample stability for longer, and we are accepting samples up to 5 days from the blood draw.
Thank you.
Thank you very much.
So next speaker is Dr. Laura Antonuzzi. She is a young investigator from the University of California, San Diego. Her title of the talk is Oxidative Stress Reprograms Epigenome to Initiate Malignant Progression via NRF2 EZH2 Self-Amplifying Loop. Thank you.
Hi, good morning, everybody. Thanks so much for this great opportunity. Although more than 90% of pancreatic ductal adenocarcinoma is initiated from a premalignant lesion known as pancreatic intraepithelial neoplasia 1, only containing a mutation in KRAS, the most common mutation is the G12D. Such lesions are present in more than 70% of individuals that are 70 years and older, most of whom never develop pancreatic cancer. So the point is how preneoplasia progress to full PDAC and what are the processes behind it. Pancreatic cancer has a multi-step etiology driven by interaction between genetic and environmental stressor. Common feature of many of pancreatic ductal adenocarcinoma risk factor including tobacco smoking, alcohol abuse, diabetes, obesity, is the induction of oxidative stress, defined as imbalance between generation and elimination of reactive oxidative species. We have identified two major players. The first one is NRF2. I don't know how much you are familiar with it, but NRF2 is the key activator of the antioxidant system and oxidative stress sensor. In normal healthy cells, NRF2 is very important because it is upregulated transiently and can be transferred to the nucleus where it activates transcription of antioxidant response gene, detoxification, and immunosurveillance. In this way, NRF2 is working as cancer prevention. So this is the reason why NRF2 was also target for chemo prevention. But NRF2 has also a dark side, and our group and many others has identified NRF2 to be upregulated in a cancer cell, driving transcription of gene involved in cancer growth, metastatic dissemination, and drug resistance. The second player is EZH2. EZH2 is a subunit of the polycomb complex repressor 2. It possesses a set domain and catalyze methylation of histone H3K27 involved in chromatin compactation and gene transcription silencing. But also EZH2 has a different aspect. It can work in a PRC2 dependent fashion, activating gene transcription together with a subset of transcription factor. To investigate oxidative stress-induced downstream mediator of PANIN to PDAC progression, we have used the experimental model of pancreatitis-induced PDAC progression. We injected mice with cerulin, wild type, and KRAS G12D mutant mice, and we analyzed the pancreata at 3 and 7 days after last cerulin injection. We analyzed EZH2 and NRF2 and downstream target. And what we observed was a transient upregulation of EZH2 and NRF2 At day 3, time point where during which we had a spike and a strong damage of the pancreas with pancreatitis. And then at day 7, when the homeostasis of the pancreas was restored, NRF2 and EZH2 were down. What was happening instead in KRAS mutant mice was that we observe an enduring upregulation of both the two proteins, correlating with a progression of the disease through more aggressive lesion. Questioning if NRF2 can directly activate PANIN-to-PDAC progression, in collaboration with Genentech, we obtained some mutant mice harboring E79Q mutation that was identified in lung cancer patient. We crossed these mice with PDX3 or KRAS G12D mutant mice, and only in this context, NRF2 was able, activated NRF2 was able to drive PANIN-to-PDAC progression. These mice harbor very early acute pancreatitis with a strong stroma reaction, and we observe a decrease in survival rate, as well as upregulation again of EZH2 and NRF2 and their downstream target. If NRF2 and EZH2 regulate each other, they form an autoregulatory loop. Questioning if that was the case, we have performed an in silico analysis of EZH2 promoter and identify an ARE binding site upstream. We knock out through CRISPR-Cas9 this single binding site, and we observe that EZH2 and NRF2 at their downstream target were again downregulated both at protein and RNA level. And the CRISP of this single binding site was enough to decrease the tumorigenic potential of human pancreatic cancer cell line MIA-PaCa. On the other side, to look as EZH2 could regulate NRF2, we also knock out EZH2 in mouse pancreatic cancer cell line derived from by all the KC mice. We perform ATAC-seq and CPT-seq, and we observed that chromatin, we had chromatin compactation, the promoter and the enhancer of NRF2 promoter, as well as loss of activation marker H3K27 acetyl. To identify targetable downstream signaling of the EZH2 and NRF2 autoregulatory loop, we perform transcriptomic analysis, and we analyzed by RNA-seq pancreata from our transgenic mice, NRF2 activated KRAS, as well as serolin-treated KRAS mice. And consistently in both model, we observe upregulation of oxidative phosphorylation, E2F target, MYC target, as well as fatty acid metabolism and adipogenesis. And through targeted RNA qPCR, we define SRBP2, FASN, HMGCR, MAE1, and CPT1A as a target of EZH2 and NRF2. In conclusion, many of pancreatic risk factors like cigarette smoking, pancreatitis, and alcohol abuse induce oxidative stress at the cellular level. And together with oncogene, like KRAS mutation, they drive PANIN-to-PDAC progression. They activate this autoregulatory loop in the cell involving EZH2 and NRF2. EZH2 can work in PRC2 dependent and independent fashion. As PRC2 complex, EZH2 inhibit acinar identity gene, cell cycle regulator, and inflammatory response. On the other side, EZH2 can activate in PRC2 independent fashion E2F target, fatty acid metabolism, and adipogenesis, driving a metabolic reprogramming of cancer progenitor that acquired a metabolic adaptation, enabling them to survive the nutritional stress that is a characteristic of pancreatic cancer and this huge stroma. And with that, I want to thank all my collaborators, in particular my mentor, Dr. Michael Karina, who supported me during my postdoc, all the founding, and in particular, the American Pancreatic Association that awarded me the Young Investigator for this year. Thank you. I will take any questions.
Thank you. Questions from the audience, please.
Thank you for a really great talk, Minoti Apte from Sydney. So I was wondering if it is oxidative stress that's mediating that progression of PANINs to, in your case, mouse.
Yeah.
The question is, sorry. So you use cerulean as the trigger, and instead of that, would you be able to use smoking or something that's really relevant clinically to drive the progression? And the second is, would you, if you treat it with antioxidants, would that prevent the progression?
We have not treated them with the antioxidant. Actually, this is also the major point. And when I show the NRF2 introducing a slide, the point is NRF2 has been targeted for chemo prevention, but we need to be very careful because when we can do that in the patient, what if the patient has KRAS already mutant? So the point is we have actually inhibited NRF2 during a cerulean model. In particular, I wait that there was enough time to have cancer progression. And when we treat with the NRF2 inhibitor, what we observed was a restoration of acinar compartment, and we were able to block PANIN to PDAC progression. So our therapeutic approach would be mostly to target this metabolic dependency of a cancer progenitor. And this is the reason why I did not have enough time to show you, but actually FASN is one of the major mediator for metabolic reprogramming of cancer progenitor. And this will be our next step for research.
I'll take one more question.
Hi, my name is Maria Sabatini from Augusta University. I have a question about the upregulation or the upstream regulation of the NRF2. I just wonder if there is a direct activation of the superoxide or involve, activation of a MI kinase like a P38 MI kinase. Can you explain a little bit more?
Yeah, thank you so much for asking that. So NRF2 has been shown already to be upregulated at the transcription level by KRAS oncogene. But what happened is that when you have oxidative stress, there is this strong upregulation, not only at transcriptional level, but also as protein stability. When you have oxidative stress, NRF2 is released by its negative regulator KEAP1, so you have protein stability and further upregulation. So on the other side, when you have oxidative stress, I could not show the data. We observe that there is upregulation of the MAP kinase SIG pathway. And this is also an essential mediator to have this huge boosted upregulation of the two, EZH2 and NRF2.
Thank you. Thank you for
so
the
much.
presentation.
Thank you.
The next speaker is a Dr. Des Wei Hill. As a young investigator, the topic from the University of North Carolina, Chapel Hill. The title of the presentation is a methylation-based classifier for pancreatic cancer subtype.
Morning, thanks. How do I move the slides? This one, oh, right. Hi, thanks everyone. My disclosures. So our lab has previously identified some tumor-specific molecular subtypes, the basal-like and classical subtypes, which correspond to prognosis, with the basal subtype having significantly lower survival rates than the classical subtype. And these subtypes also are predictive of chemotherapy response. This can be seen here in this waterfall plot of some results from the COMPASS trial where patients with classical-like tumors have better response to chemotherapy treatment compared to patients with basal-like. And this subtyping schema that you see here was done with the purest classifier. It's currently in clinical trial, and it's based on a gene signature of gene expression, which requires RNA-seq analysis of tumor tissue. So it requires a biopsy. So if we were able to classify subtype based on a marker present in blood, that would allow us to subtype pancreatic cancer using a less invasive assay like a blood test, which is why we're looking at DNA methylation as a marker. We already know that there is differential signal between the subtypes in several data sets, including the TCGA data set, the Bailey et al. Data set, as well as some of our own new patient-derived xenograft tumors. And then also, if we can get the signal found in the bloodstream, we should be able to do a liquid biopsy subtyping test for subtype based on methylation. So we've trained a new classifier, which for now we're calling Cosmos, and it predicts subtype based on the methylation status of a set of sites. And we've used Purist as our ground truth subtypes. TCGA was used as our training data set, and the Bailey et al. And our PDX were used as two completely different independent validation sets. And these are all array sets. And the random forest, this is an ensemble method. It involves constructing a collection or forest of decision trees. And each tree has the sample follow a certain path based on the values of the different variables through that tree. And then each tree votes on whether the sample is basal or classical. And the overall classification is based on a majority vote of the trees in the forest. So after performing an initial probe selection using 5,000 nominally differential probe sites and removing some problematic probes from the array, like those which are liable to cross-validation or those where the probe has a common SNP, we arrived at a set of probes to start training and tuning our random forest. And using a k-fold cross-validation approach and tracking variable importance throughout these iterations, we were able to arrive at a set of probes which were not only predictive based on random forest important measures, but also stably predictive over lots of iterations of random forests. And then we used the k-top scoring pairs method to narrow down our variable selection to arrive at 20 stably predictive sites for subtype, which you can see over here. So these are our 20 sites. Half of them are highly methylated in basal samples, half are highly methylated in classical. And we, using the Bailey set as a first validation, we achieved good prediction accuracy with an AUC of 0.98 and perfect prediction in our own set of patient-derived xenograft samples. And our subtyper also had good correspondence with previous subtyping schemas corresponding with the Bailey et al. Squamous subtype, the Moffitt basal, the Purist basal, and the Collison quasi-mesenchymal subtypes. And we've also not only reproduced, but slightly improved upon the significant difference in survival from the Purist classifier. We're working on getting probes designed, methylation-specific PCR probes for these sites. And the preliminary results are looking quite nice with the qPCR-based methylation values corresponding well to the EPIC array-based methylation values. So this is looking promising to translate this into a qPCR and then hopefully a ddPCR-based assay, which we can then move into working with plasma. So moving this to a liquid biopsy. The next question we wanted to ask is whether there were any functional relationships associated with these predictive probes. So we looked at genes whose expression was significantly correlated with the methylation level of these probes, and found, especially in the set which was of genes, which was positively correlated with the basal probes and negatively correlated with the classical probes. So one could perhaps interpret this as genes which are potentially downregulated or repressed in classical tumors and upregulated in basal tumors. We found some enrichment related to functions of cell adhesion and invasion. So we wanted to see if we could get some biological evidence for this. So we performed an siRNA knockdown using 2 separate siRNAs for Rep15 in a classical cell line. This is using an invasion assay where you load your cells into a transwell, and after an incubation, you count the number of cells which are able to migrate through this membrane. And we found significant increase in invasion in our 2 siRNA knockdowns compared to the control siRNA after 48 hours of incubation of Rep15. So we're moving forward to test some more invasion and other assays of these predictive probes. So in summary, we've developed a methylation-based classifier for PDAC subtype. It's using only 20 methylation sites. We validated it on 2 independent data sets. It achieved 100% accuracy on our in-house patient-derived xenograft tumors. We're working on designing methylation-specific primers to test this currently with qPCR, moving towards ddPCR to be able to detect the smaller amounts of DNA that one would find in plasma. And hopefully this can become a less invasive, easier, and clinically applicable subtyping assay. And thanks to the Yale lab.
Thank you. So questions from the audience?
So very nice work. This is Manish Jain from University of Nebraska Medical Center. So when you talk of the classifications, and we know now that these subtypes are not locked into basal classical, there's actually now a gradient with overlapping things. So how do you see your scheme? Like, did you, when you were categorizing, did you see an overlap, some tumors which are fitting in both types? That's question number 1. And question number 2 is, how is the status of metastatic disease going to cloud your decision on going to the subtype based markers if you don't see the same subtypes in the metastatic disease?
So I think the first part of your question, did we see more of a gradient as opposed to just basal like or
It's a
classical?
gradient or an overlap, like certain
Yes.
samples not fitting in either or fitting partially in both.
Yes, so the classifier in the same way that Purus does, you can see samples that are not 100% basal looking or 100% classical. So you will get a gradient. For the purposes of a clinical decision, the classifier does a cutoff and says mostly basal or mostly classical. But yes, absolutely, this is not a 2-box problem. This is a spectrum. And you had a second point about.
So what about the metastatic disease when you're planning to go for a blood-based classifier scheme? And if there's already an occurrence of metastatic disease, so, and if the subtypes don't follow the same pattern there?
Yes,
So
that's a good question. I haven't tested this on a bunch of metastatic samples yet. So we would have to look at that.
Thank you.
I have one question for you. So you used like 82 ICGC samples and 21 PDX tumors to get this data, correct?
Those were the validation.
Validation part.
Yeah.
How many samples did you use initially?
To train?
Train.
150 TCGA samples.
So my question is, it's kind of like, we discussed this yesterday, one of the sessions. So how many tumors do you think will be needed to get like a clinically relevant biomarker system? If, like, for example, in order to get the appropriate statistical power to show that, yes, this can be a biomarker for the future. So how many do you think tumor samples will be really needed for statistical power? Approximate. You don't have to get to 2,521, but
okay.
Yeah. It's statistical power we're talking. So
yeah. And that's going to depend on the biomarker and depend on the general range that we're going to see in that biomarker, I think. But I would agree.
A lot more.
Yeah.
Okay. All right.
A lot.
Thank you so much and nice work. Thank you.
All right, we're going to move on. We've really had a nice diversity of talks. We're going to probably talk about our first myeloid focused talk. And the presenter is going to be Dr. Dixit from the University of Minnesota, who's also a recipient of the Young Investigator Travel Award from the APA. His talk is entitled Blocking TNF-Producing Macrophages Activates Anti-Tumor Immunity in Pancreas Cancer.
Good morning, everyone. First of all, I would like to thank APA for giving me this opportunity to discuss my work. Today, I'm going to talk about some aspect of my work which deal with macrophages. As we know that macrophages are essentially involved in each and every stuff in pancreatic cancer. They are involved from initiation, migration, drug resistance, and immune suppression. So far, what we know about macrophages, they can be either M1 type or M2 type. They can come from bone marrow or they can come from resident ones. Besides that, we have very limited information in pancreatic cancer. So the goal of this study was to get a comprehensive understanding of macrophages in pancreatic cancer. So we began with analyzing publicly available single-cell sequencing data from the FT lab. And what we found is like this macrophages are actually, they don't form very tight cluster. Like each colors actually represent macrophages from individual patient. And so we looked at different markers, like what is the distribution of these markers. On the top panel, you can look at the ontology marker, which actually tells like what's the source of this macrophages. And the first part is like resident one, MERTK and CX3CR1. And what we found is like, not like if you look at the left side of panel, like those patients that have very low expression of these markers, whereas the right side, they are pretty high. And what we found is like when you look at the markers of bone marrow-derived macrophages, they are pretty abundant, they're pretty high. We also look at the distribution of genes related to immune modulation. And what we found is like, there's no consensus. Some markers are evenly distributed, some are only present in few patients. And so we began asking, how should we target this? So we thought like, since most of these macrophages, they represent like they express pretty high bone marrow derived marker, let's start with targeting bone marrow the macrophages. To understand that, what we did, we actually enrolled KPC mice in a preclinical trial setting, and we took KPC mice which has 48 millimeter tumors, and we enrolled them in different groups. Like we had group gemcitabine, and immune checkpoint blockade. And we use one group which has gemcitabine, immune checkpoint blockade, which is like PD-1, CTLA-4, and CCR2 inhibitor. For this, we use a CCR2 inhibitor, oral inhibitor, which was developed by a company, Chemocentrics, California. And you can see like when you use the triple therapy, you see a huge decrease in macrophages population. You also see a huge decrease in M2 macrophages, and you also see a huge decrease in MDSs, which says that like if you inhibit macrophages, you're able to inhibit MDSs and macrophages as well. And what we also find like in the triple therapy, when you compare with gemcitabine or dual therapy like gemcitabine and ICB, like immune checkpoint blockade, the triple therapy showed higher amount of CD T cell migration into the tumor, and they showed decrease proliferation and increased cell death. If you also look at the meds are pretty notoriously difficult to treat. So we decided to look at like what happens in these mice. When you look at the meds, these mice, like mice treated with triple therapy, has significantly reduced liver and lung metastasis. They also had this metastatic lesion. They also have very high, significantly high CD8 T cell in the lesions. And we also see increased cell death in response to triple therapy. We were like curious, how does it happen? What's the mechanism behind this? To address this question, we did the cytokine array analysis of gemcitabine and gemcitabine immune therapy treated and CCR2 inhibited tumor. What caught our attention was like IL-33. It was hugely, very significantly expressed in triple therapy. And when we did IC, we found like this IL-33 is actually highly expressed in cancer cells. And IL-33 kind of have like a dual role. It can be pro-tumorigenic or it can be anti-tumorigenic given the context. So just to see what's happening in our system, we took KPC cell lines and implanted with recombinant IL-33. As you can see in the blue bar, like cells implanted with IL-33 grew pretty slowly. And when we looked at the tumors, this samples has pretty high CD8. What this tells is like IL-33, at least in our system, in our setting, able to induce immune response, anti-tumor immune response. So we were curious, like how macrophages and IL-33 are connected. When we inhibit macrophages, we see huge increase in IL-30. To address that, we went back to our original single cell sequencing data from the human. And what we saw is like all these macrophages, they have pretty high TNF-alpha signaling pathway. And we validated this further in KPC tumor by dual staining TNF-alpha and F480. And what we found is like most of this TNF-alpha comes from macrophages. And if you were to knock, like when we knocked out KPC, CCR2 in KPC model, which is actually required for recruiting bone marrow derived macrophages, we see a huge decrease in TNF-alpha. Similarly, we saw decrease in TNF-alpha in triple therapy treated group. So what it tells us is like, most of this TNF-alpha comes from macrophages. And so we're like, how this TNF-alpha actually, how does it actually, does it regulate IL-33 expression? So address that, we took KPC cells, we treated them with TNF-alpha, and we measured IL-33. What we found is like, TNF-alpha treated cells, actually KPC cells shows decreased IL-33, both at gene level and protein level. So in summary, what we showed in this study, what we found in this study is like, when you have in tumor microenvironment, you have ICAF, MICAF carcinoma cell, there's a huge heterogeneity in the ability to recruit myeloid cells. My ICAF secrete highest amount of CCL2 in tumor microenvironment. So their ability to induce immune suppression is pretty strong, followed by MICAF, followed by carcinoma. And carcinoma cells also, metastatic cells secreted a higher amount of CCL2 when compared to the primary tumor cells. And in unperturbed tumors, when you have high myeloid cells, you have high TAM, you have high TNF-alpha, this actually decreases IL-33. And what we believe is like when these cells die, they actually induce silent death. So there is no immunological response to this kind of a death. However, but if you block CCL2 and in turn you block myeloid cells recruitment, these cells has lower TAM and they have higher expression of IL-33. When these cells die, they actually induces, like IL-33 is alarming. So when these cells die, they increases they increases like immune response, and this death actually causes more T cell activation via DC and ALC2 cells. So I would for this, like of course, I would like to thank my lab and other people who like involved in this study. And we also especially like to thank Rolf Brecken and Hugh Kong, who were like really involved in single cell sequencing data analysis. Thank you.
This paper is open to the floor.
Can you hear me? Yeah.
Uh-huh.
So I had a question in the CCR2 knockout or CCR2 inhibition. You mentioned that there's a mixture of resident and bone marrow derived macrophages. So my question is I would expect that CCR2 deficiency would prevent the influx of bone marrow derived macrophages. When you knock out or inhibit CCR2, do you see selection in the tumor microenvironment for a different kind of macrophage that might be undergoing proliferative self-renewal within the tumor microenvironment?
That's a very good question. Thanks for asking that. So the idea behind this study was like, if we delete bone marrow derived macrophages, do resident macrophages actually take over? Do they become more immunogenic? Like what we know that resident ones are more involved in fibrogenic activity and bone marrow derived are immunogenic. So we haven't done like any single cell sequencing to answer that question. But what I believe based on the data is like they do not take over. Yeah, so they have specific functions.
Thanks.
What what Dave Linehan's group has shown in the clinical samples is there's a compensatory myelopoiesis with CXCR2 expressing granulocytes. So I wonder if you've in sort of piggybacking on his question, whether you've seen a compensatory increase in granulopoiesis.
Yeah, that's a good question. So in KPC model, we don't see it. Like if we do use orthotopic, we do see some, but in KPC setting, no, not at all.
And do you think that TNF in the macrophage has something to do with the ontogeny of fate determination between M1 and M2?
That's a good question. To be honest, we don't know. We need to like do extra experiment to address that question.
Right now,
Oh, there's one other question.
Yeah, sorry, I have a quick question. So in your study, did you see any changes in like stroma or inflammatory castes were going down along with like upregulation of macrophages?
Can you say that again?
Did you see any changes in stroma or like a stromal component along with like changes in inflammatory castes
Okay.
in your knockdown?
So we did like fibrogen collagen analysis. We did alpha SMA. That's what we looked at. We don't find any significant difference. So what we believed is like, since they still have resident macrophages and they are involved in fibrogenesis, so they are doing their job. So once we block bone marrow-derived macrophages, we are specifically blocking like immunosuppressive macrophages.
Thanks.
Thank you very much. We have to move on.
Thank you.
We're really loving the audience participation, and we're going to completely switch gears and talk about a clinical topic next. Dr. Fogwe from the Mayo Clinic is going to be presenting his talk, which is entitled Endoscopic Ultrasound Guided versus Percutaneous Drainage for the Management of Postoperative Pancreatic Fluid Collections after Distal Pancreatectomy.
So thank you, everybody. I want to thank the APA for this opportunity to talk about our study. And we'll be switching gears here, hopefully taking the audience closer to the bedside. So postoperative pancreatic fluid collection remains a significant source of morbidity after distal pancreatectomy. A lot of these patients would develop symptoms such as nausea, vomiting, or abdominal pain, even infection requiring some form of drainage. In the past, percutaneous drainage was a mainstay of management, but with the advent and success of endoscopic drainage technique in the management of inflammatory pancreatic fluid collection, some of this intervention has been used for the drainage of postoperative fluid collection. There have been a few published studies on this, but they've been grossly limited by limited sample sizes and also like heterogeneity in the pancreatic resection technique and fluid collection type. So as you can see, we have two really nice images on the board. The first one is a patient with pancreatic neuroendocrine tumor, underwent distal pancreatectomy, develop a fluid collection within the distal pancreatectomy resection bed. This fluid collection aborts the gastric lumen, which can be easily assessed both by percutaneous and endoscopic drainage technique. In the second case, the patient with history of a pancreatic ductal adenocarcinoma underwent a pylorus sparing Whipple's procedure. And as you can see, develop a fluid collection within the pancreatic resection bed in the head of the pancreas, and also some issues with delayed gastric emptying. So as you can see, lumping both cases together, and even in patients with fluid collection that are lower in the pelvis, becomes difficult to interpret the data. So the main primary outcome of our study is to compare the clinical success between percutaneous and the endoscopic drainage techniques after distal pancreatectomy, and secondary outcomes include like technical success, total number of interventions, time to resolution, rates of adverse events, and also fluid collection recurrence. So this is a retrospective study from a single academic center spanning over a time frame of about 9 years. We included adults only who underwent distal pancreatectomy and developed fluid collection just within the distal pancreatectomy resection bed. And we excluded anyone that had other form of surgery as not distal pancreatectomy, so like total pancreatectomy, pancreatic enucleation, or even a Whipple's procedures. Patient with fluid collection that wasn't symptomatic, we didn't include in our study. And also patient with fluid collection that was outside the pancreatic resection margin was also excluded as well. So this is a flow chart looking at the data that we reviewed. Majority of our data came in from a prospectively maintained pancreatic database, kind of supplemented by an electronic medical query. After excluding duplicates, excluding patients that were outside the study period, also excluding patients that were outside that didn't have like the pancreatic resection surgery, we ended up with about 1,046 cases. And after excluding patients that didn't have any fluid collection or patients that had fluid collection that were outside the resection margin, of course, we ended up with about 455 cases. So after excluding patients that did not have any fluid, that had fluid collection but didn't require any intervention, and patients that we didn't have follow-up data, we ended up with about 217 patients in our study cohort. And of this, 106 was endoscopic ultrasound guided cases, while 111 were percutaneous cases. I also like to highlight that of the ultrasound guided cases, 36 were lumen apposite metal stent only, 43 were double pigtail stents, while 27 were lamps with coaxially placed double pigtail catheters. So we define clinical success as symptomatic improvement and radiographic resolution of the fluid collection to less than 2 centimeters without the need of an alternate drainage modality. And we define an intervention as any procedure that involves a stent or catheter placement, removal, adjustment, or even reinsertion as well, and even necrosectomy. In cases of patients that underwent like catheter placement via percutaneous drainage that had it removed in the clinic, we did not count that as an intervention. So fluid recurrence was any recurrence of symptoms or new fluid collection on the cross-sectional image within 6 months post a procedure. So I would like to say that patients that had fluid recurrence were not counted as a clinical failure. So these are data of our baseline characteristics. I would like to draw your attention to a few numbers here. As you can see, majority of the patients that underwent percutaneous drainage underwent open surgery when compared to the endoscopic drainage cohort. Majority of the patients that underwent percutaneous drainage had initial drain placed while they were inpatient as compared to the endoscopic cohort as well. Majority of the patients that underwent endoscopic drainage had more necrotic tissue. And also majority of the patients that underwent percutaneous drainage had drain placed earlier, so 10 days as compared to almost a month in the endoscopic drainage group. The size of the fluid collection was the same between both groups. So just diving straight into the results. So again, the clinical success rate, which is the primary thing we're looking at, was statistically significant favoring endoscopic drainage. So remember, we had about 28 patients that we didn't have follow-up data for. 21 of these were percutaneous cases and 7 were endoscopic ultrasound cases. So in order to account for them, we did like a sensitivity analysis, initially assuming that the patient lost to follow-up with clinical failures and also assuming that there were clinical success. As you can see in both cases, the results is about the same. So secondary outcomes look like a fluid recurrence and also like drain removal. As you can see, recurrence rate was lower in the endoscopic drainage group and the drain dwell time was longer in the endoscopic drainage group, and both were statistically significant. Also, we looked at the number of interventions. As you can see, it was higher within the percutaneous group, and this is the number we have there is a median and lower in the endoscopic drainage group. But I'd like to highlight that majority of or some of the cases in the percutaneous group were just what we call sinograms or drain checks without necessarily drain exchange. So most of these were simple and quick procedures that did not require sedation. So here's a table of our adverse events. There wasn't any significant noted, but as you can see, a majority of the adverse events within the endoscopic group were stent migration. So most of these were they actually placed double pigtail catheters, like migrating to the small bowel, causing issues with bowel obstruction, while within the percutaneous group, it was infection. So we believe that one of the merits of our study is that this is by including just patients with fluid collection within the distal pancreatectomy resection bed only, it allows for a fair comparison between both groups and also decreases heterogeneity within the study cohort. Additionally, I think it helps to increase the external validity of our study as well. Endoscopic drainage were performed later. We believe that this may partly be due to the fact that these techniques are coming from patients with inflammatory fluid collection. So the guideline states that wait for almost a month, 4 weeks for this wall to mature. So that may be one of the reasons why it is performed later. Even though there have been some recent studies on this, including one from our group that says that you can pursue this drainage as early as a week after surgery without any change in the adverse events or clinical outcomes. Endoscopic drainage was used more often in patients with solid necrosis, and higher recurrence rate in the percutaneous may be related to the fact that the drains were removed earlier. So limitation, there was a retrospective study. Again, the patients were not randomized. Another additional aspect to note as well is that crossing over from like percutaneous to endoscopic wasn't necessarily a clinical failure. It may be a clinical decision based on the case and based on the patient's or provider preference. Again, this is a single academic study, so it's difficult to generalize the results. And we didn't account for patients that had concurrent fluid collection. For example, you may have percutaneous cases that had fluid collection within the distal pancreatic resection bed that's being drained, but they may also have fluid collection in other areas that are also drained concurrently as well. So with this, I'd like to summarize that endoscopic ultrasound guided drainage was associated with higher rates of clinical success, lower need for re-intervention, lower rates of collection recurrence, and longer stand duration. So we'd like to conclude that in centers that have this ability to perform this procedure, they should consider using this technique as a definite treatment for fluid collection within the distal pancreatectomy resection bed. Thank you.
This abstract is now open to the floor, and I was remiss in mentioning that Dr. Fogwe is also a recipient of the Young Investigator Travel Award from the APA.
Good morning, Carlos Fernandez del Castillo from Boston. As a surgeon involved in distal pancreatectomies, I'll say this is an unsolved problem in surgery. I disagree with your conclusion because one could leave here and saying the way to drain this is always endoscopic. You drain endoscopic 4 weeks or 3 weeks after, percutaneously a week after. A patient 5, 6 days after a distal pancreatectomy who has fever in a fluid collection, you're not going to drain that endoscopically because that collection is not mature enough. You're going to create more problems. I think the message is we have two tools. We have to be selective when we use them. If I'm a patient, I'd rather have an endoscopic procedure. Of course, I don't have a drain outside, but I don't think we can freely say or recommend that endoscopic drainage is the way to go.
Yeah, I think that is really a valid, concern and question. This is something that was brought up in our group as well. Remember, we have advanced endoscopists, interventional radiologists, and also our pancreatic surgery group that was involved in this study. And they also brought out that valid question as well. How can you compare like an endoscopic drainage with percutaneous drainage? Sometimes it's difficult to compare both. But again, going to your question regarding the fact that, most of the percutaneous cases were done early, there is data showing that we can do this earlier. I think we have an earlier study that came in as part of this study that proved that. So that aspect of early drainage can be done.
I don't doubt it.
Yeah.
But then you would have to show patients who had early endoscopic drainage versus patients who had early percutaneous drainage. And I don't think your data is going to be as favorable because there's going to be some potential disasters.
Correct.
All right, in the interest of time, we have to move forward.
Thank you.
Thank you.
The next presenter is Dr. Sandeep Suwain from Duke University. His title is Pressure Sensing Piezo 1 Channel Signaling Accelerates Pancreatic Cancer Growth and Metastasis.
Good morning, and thank you, APA, for this nice opportunity. So if you see, the PDAC is one of the aggressive form of cancer and notoriously difficult to treat. The dense desmoplasia surrounding the cancer cells is the key feature of the PDAC, which accounts for 90% of the total tumor volume. And if you see, during PDAC, the activated stellate cells produce inflammatory cytokines, which can promote PDAC growth and metastasis. And same time, the activated stellate cell produce the extracellular matrix protein like fibronectin and collagenase, cause the stroma, dense stroma. Recently, we discovered that pancreatic stellate cells are highly sensitive to pressure by expression of a mechanical ion channel PIZO1 and TRPV4. And if you see in tumor, these channels can be activated by mechanical force or high pressure or shear stress. In tumor, when the cell grows, the surrounding tissue or cells get high pressure. So it is obviously that channel PIZO1 may be activated in the stellate cell macrophage or endothelial cells. So recently we reported that in pancreatic acinar cell and stellate cell, there is a high pressure mediated intercellular calcium rise through PIZO1 channel can activate phospholipase A2 enzyme. That enzyme breaks down to phospholipid to arachidonic acid, and arachidonic acid derivatives like epoxy epocytonic acid, which are the activator of a TRPV4 channel, open the TRPV4 channel and cause excess calcium inside the cells. That calcium is cause activation of pancreatic stellate cells. So this mechanism observed in the chronic pancreatitis or pancreatic fibrosis during pressure-induced chronic pancreatitis, we observed that when we ligate the tail region of pancreas up to 8 days and 30 days, there is an increasing intercellular calcium because of PIZO1 TRPV4, and that lead to the activation of pancreatic stellate cell and cause pancreatic fibrosis. So once in PDAC, once the stellate cell get activated, it produces inflammatory cytokines. And if you see, the IL-6 is the key inflammatory cytokines, and it promotes PDAC growth and metastasis. And IL-6 is the key mediator of STAT3 signaling in hepatocyte. And once STAT3 activation in hepatocyte, it increases the production of acute inflammatory proteins like serum amyloid 1A and serum amyloid A2, and which favors the accumulation of myeloid cells and promote the metastasis. So if IL-6 and STAT3 and SA the key signaling factor for metastasis niche, and we know that is activated stellate cell produce IL-6, and PIZO1 is responsible for activating pancreatic stellate cell. So we thought that study the role of PIZO1 in PDSC growth and metastasis. So to develop, we knock out a genetic mice model, tamoxifen genetic mice model, we knock out the PIZO1 in the GFAP expressing cells, glial fibrillary acidic protein cells, mostly marker for stellate cell, and I use the tamoxifen for 5 days, and I use the mouse 7 days after last tamoxifen injections. So in this mouse, to develop the PDSC, we injected the KPCY cell orthotopically to the tail region of pancreas. And after 30 days, we collected the pancreatic tissue and the serum and liver and studied the analysis. And if you see here, the genetic deletion of PIZO1 in pancreatic stellate cells reduced the tumor growth and significantly reduced the pro-metastatic niche factor, right? IL-6, SA1 and SA1, SA2 in together. So if you in PDSC, we observe that there are two types of CAP mainly characterized. One is ICAP, one is MyCAP. The ICAP is people describe that there is low in number, high expression of a smooth muscle actin, present close to the neoplastic cells. And ICAP is the high in number, express more IL-6 and IL-11 and LEAP, and present distant to the neoplastic cell. So we found that PIZO1 agonist ODA1 at higher dose activate human pancreatic stellate cell to ICAP phenotype. If you see this one, the pancreatic stellate cells are cultured in the matricial coated plate to maintain the coefficient, and you see the blue dots are the fat droplets surrounding the stellate cells, and upon ODA1 application, it loses the fat droplet, and in 24 hours, we find the increasing expression of mRNA, of fibronectin, and increased expression of IL-6. Additionally, observe that ODA1 dose dependently reduce the smooth muscle actin expression in human pancreatic stellate cell and increase the fibroblast activation protein. If you see, all these are the characteristic feature of ICAP. And we recently people reported that CD10 positive pancreatic stellate cell are promote the PDSC growth and metastasis. And in our experiment, we observed that ODA1 dose dependently increase the CD10 expressions. So if you see here, if there is high pressure in the PDSC or stiffness, they may activate PIZO1 in the stellate cell and cause pancreatic stellate to ICAP phenotype rather than MyCAP phenotype. So I described that a TRPR channel operates downstream of PIZO1 signaling, which is responsible for pressure-induced pancreatic fibrosis. Then we studied that if to determine the role of TRPR in PDSC, we injected the KPCY cells into the tail region of pancreas in TRPR knockout mice and wild-type mice. As you see here, in TRPR knockout mice, there is a decreasing pancreatic tumor size comparatively, and also decreasing the inflammatory cytokine markers like IL-6, SA1, and SA2. So also we, because of time limit, I didn't present the metastasis study, but also we found that in TRPR knockout mice, there is a reduction of metastasis lesion and size of metastasis and frequency of number of metastasis in the liver is reduced in the TRPR knockout mice. So in summary, you can conclude that pressure sensing PIZO1 channel, mediate TRPR channel, activate which can accelerate the pancreatic cancer growth and metastasis, and possibly blockage of TRPR channel may reduce PDSC tumor growth or metastasis. Thank you. And I like to thank you, my mentor, Dr. Rajul Reddy, I working with Dr. Rajul Group, and my colleague Joel Romack, and NIH grant. Thank you. And Duke University.
Thank you. Questions?
Hear me? Okay. So I'm a student from University of Nebraska Medical Center. So I just attending journal club recently. So this molecule or this target you're working on is also associated with left ventricle hypertrophy, which is a heart disease. So have you monitored or have you see any heart kind of thing with the mice model you are working on?
In TRPR knockout or PIZO knockout, we didn't observe that heartbeat, but the mouse looks fine. The behavioral no changes and no weight change in the mice. But we never check the heartbeat or heart hypertrophy measurement. I never checked. Yeah.
Okay, thank you.
Hi, Jason Bruce, University of Manchester. So fantastic talk, really nice data. Forgive me if I missed this from the beginning, but is piezo-1 expressed in the cancer cells as well as the stellate cells?
Yeah.
And does it have a role there?
Yeah, so we check the piezo-1 expressed in the cancer cell, the like PANC or the other cells, but the expression is quite low. The expression level compared to stellate cell is much lower there. And I studied the calcium imaging, we didn't get that much calcium rise as expected in the stellate cell. And piezo-1 and TP4 both expressed in the macrophage and endothelial cells. That could be reason that we TP4 knockout mice, you get much better effect than the piezo-1 knockout in the stellate cell mice. So because piezo-1 is knockout in the stellate cell, macrophage and endothelial cells, so maybe the inflammatory response also, these cells also sense the pressure. So that's why we're suggesting that TP4, which mouse is better response than the piezo knockout mice.
Okay. Just one very quick question again, if that's okay. So is the piezo-1 directly sensing the pressure within the
Yes.
stellate cells, or is there any interaction with any
No, the
extracellular
piezo-1
matrix?
inherently mechanism-sensitive, so they have the domains, they can sense directly pressure.
Okay, thank you.
Thank you.
Have you seen piezo-1 or TRPV4 upregulation in clinical metastatic samples?
In vitro, we studied the human stellate cell when I applied the shear stress or one. We increased the piezo-1 and the tripper, but in clinical mouse tissue, we didn't check it. In mouse pancreas, we didn't check it. Yeah.
I have a
Okay.
I have a quick question. So how do you reconcile? So you obviously used a stellate cell marker, but how do you reconcile this data with some newer data that's coming out looking at FABP4 as a stellate cell marker and showing that it only accounts for a minor subset of tissue resident cancer associated fibroblasts? Have you looked at fibrocytes,
Yes.
traveling from the bone marrow, for example?
Yes. So I think that is a good question because it is difficult now that the subtype of fibroblast and resident micropar. But we focus mostly ICAP, MICAP, but maybe there is different subtype of ICAP and subtype MICAP still is working on. So when you isolate the human pancreatic stellate cells, we found all the cells have the perinuclear fat doublets and the expression of a fibroblast activated protein and expression of GFAP. So we use these three markers. Maybe we have to go for a maybe single cell sequencing, other marker may be required to study the subtype of ICAP in the future. Yeah.
Thank you so much.
All right, we're going to move on to our next talk. Yeah, as you can see this from this rich diversity of talks that we have here to satiate every scientific palate. And I just want to make one comment that we're probably going to spill over a little bit into the morning break. And I obviously know that given the scintillating nature of these talks, no one is dreaming of leaving the session early, but I just want to mention that we will be probably spilling over into the morning break. The next presenter is Dr. Shepanen. I'm sorry if I'm messing up your name. She's from the University of Helsinki and Helsinki University Hospital. The title of her talk is Anatomical Pancreatic Variants in Intraductal Papillary Mucinous Neoplasm Patients.
Good morning, and thank you very much for the opportunity to present our findings. I am a pancreatic surgeon, and Karina Johansson, who was the first author in this paper, is radiologist, but is due shortly and was unfortunately then unable to fly over. So branch duct IPMNs are a vast number of patients that we have a big loss of resources or use of resources to detect and survey them to find these very few patients that will develop cancer eventually. So it would be important to find out features that would indicate either high-risk patients or very low-risk patients. And two things that we were looking at this study were as a pancreatic and meandering pancreatic duct, which I will explain a little bit more further. This study is retrospective. We looked 214 patients altogether. 108 had branch duct IPMN, and 106 patients had an MRI done for other reasons, for example, bile duct stone suspicion. And the pancreatic duct was analyzed in the head of the pancreas area. there are non-meandering main pancreatic duct. Most typically, the shape is descending, but it can be steeper and called then vertical or sigmoid. And the meandering pancreatic duct types have typically been these loops, either up or down, or then reverse Z types. And the new finding or observation that we made was the N shape. And also we wanted to check if that was in association with worrisome features or high-risk stigmata with these IPMN patients. Here are pictures of the first three ones are from reverse Z subtypes of meandering pancreatic ducts. And in the last picture, there's also a duct of Santorini. And here are the loop up and loop down configurations in the first two pictures, and the picture G describes the N-shape meandering pancreatic duct. And when the duct of Santorini makes a curve and connects to the main pancreatic duct, that's where ansa pancreatica occurs. And here are the results. Meandering pancreatic duct in total was more common in branch duct IPMN patients than in the control patients. And here you can see the numbers of these different subtypes. And also when we look at things vice versa, if you see a meandering pancreatic duct, you can suspect that the patient most likely may have branch duct IPMN. And when we looked at N-shaped meandering pancreatic ducts and compared them with these most common non-meandering descending pancreatic ducts, they had more often cystic mural nodule. And the patients that had these ansa pancreatica formations had often these branch duct cysts throughout the pancreas. And in conclusion, IPM patients exhibit more often meandering pancreatic duct than the control patients, and ansa pancreatica was associated with several branch duct cysts, and also the N-shape meandering pancreatic duct was associated with cystic mural nodules. So this is the first observation of this type, and may associate with these worrisome features, but obviously this will need validation data. Thank you.
Very fascinating talk. The abstract is open to the floor.
Somnath, University of Miami. Great talk. Could you maybe possibly comment on why someone gets a meandering pancreatic duct? Like, what are the causative factors, if you will?
This is a very good question. Obviously, we don't know for sure. These are considered that they are partly developmental when there's a lack of fusion of pancreatic ducts during fetal time, but we don't know if some of these are acquired during the IPMN disease.
Thank you.
Chris Wolfgang, NYU, real brief question. Professor Sepinen, that was excellent study. This, the human mind introduces bias based on our understanding of anatomy and things like that. I was wondering if you ever considered or have tried taking your imaging data and subjecting it to AI unsupervised pattern recognition, and even in this meandering duct, is there some other third feature that we're not even looking for, recognizing that is the true association with IPMN.
I think this is a very important point. AI is something that we should be having more included in our radiological analysis as many other areas too. But I think it would improve also, as we know that there are some indications that the cyst fluid analysis may help us with the diagnosis.
Yeah, that was going to be my question. Have you looked into, the correlations with cyst fluid CEA or KRAS or GNAS clonality in these particular conformations? And my second question is, do you think that these are just clinical associations, for example, uncinate duct dilation with incidence of pancreas cancer, or do you think that there is something about ductal formations that are physiologically linked with IPMN pathogenesis?
This we don't know yet. We would need bigger data and analysis of larger. This was a retrospective series, so we don't have this fluid analysis, unfortunately.
We have to move on in this time. Thank you.
The next speaker is a Dr. Jing Yang, who is a young investigator, Cedars-Sinai Medical Center. Title of his talk is CHOP DDIT3 depletion in pancreatic beta cells restrain PDAC development and metastasis in liver.
Just to clarify, I'm not Jing Yang. Hopefully he'll make his debut at an APA meeting in the future. But my task is to bring forward the story that very interesting data that we've been developing. This is to.
And you are the green button.
Oh, cheers. Didn't see that. So just to bring some background information on our interest in obesity and its connection to pancreatic cancer development. Obesity and diabetes are significant risk factors for PDAC progression and metastasis. That's well known. Obesity-induced PDAC progression was associated with fatty liver disease and hyperinsulinemia in experimental models. There's 3 of our papers there. Fatty liver disease was proposed to mediate obesity-induced cancer links, as obesity in the absence of fatty liver had little effect on the overall risk of PDAC. And finally, bariatric surgery very often resolves diabetes and fatty liver disease and reduces the incidence of pancreatic cancer by 50%. So the laboratory of Randy Kaufman at Sanford Burnham in San Diego has published this work previously, demonstrating a very interesting endocrine-exocrine connection. It really focuses on the fatty liver disease, but they made their change in the beta cell of the pancreas, affecting this transcription factor associated with extensive ER stress called CHOP. So they found that CHOP, the genetic deletion, reduced blood insulin levels and hepatic steatosis in a model of fatty liver. So they had the mice on high-fat diets. They found that the CHOP deletion in the beta cells normalized the serum insulin, you can see in the red bars, and then completely eliminated the presence of fatty liver, as you can see in the central panels, and normalized also the level of liver triglycerides. And in an unusual and interesting therapeutic approach, they brought the concept of using this anti-CHOP, antisense oligonucleotide, which could be administered to mice. And it homes to the beta cell, and I'll show you why it's doing that. It's actually bound to the glucagon-like peptide, which the beta cell expresses a good deal of this specific receptor, so it tends to home to the beta cell. So administering the ASO normalizes insulin as well, and also reduce the liver triglyceride. So we asked, this is a neat trick, based on the fact that the liver being in a fatty state may make a better site for metastasis. We want to investigate PDAC metastasis, which often metastasize to the liver. But we also ask whether it would have an influence on the primary cancer as well. This procedure of knocking down CHOP in the beta cell. So this is the experimental model. This is carried out at Cedars-Sinai. So we use the KC mice. These are expressing a mutant KRAS G12D in the pancreas. So they're fed on control and high-fat diets for 11 weeks. And then we administered the control ASO, which has no effect, knocking down CHOP, and the specific CHOP knockdown ASO for 4 consecutive weeks. And then examined the panel progression, the glycemic control, and the fatty liver in these animals. And you can see in the box a little more detail about the ASO. This is made by a company called Ionis Pharmaceuticals. Very nice reagent for knocking down a specific target. So the body weight changes were pretty much as expected. High fat diet, as you can see on the left, compared with controlled diet, produced mice with significant weight gain. And similarly, mice treated with the different ASOs, they still gain weight on high fat diets. So of course, we want to examine, first of all, is the treatment producing elevation of any specific genes? So particularly we look at CHOP. CHOP was induced by the high-fat diet and dramatically suppressed by knocking down CHOP naturally. So in accompaniment with that, the insulin level was also dramatically reduced. This is another factor that increased with high-fat diet. It's part of the metabolic syndrome. And the CHOP knockdown effectively reduced pancreatic levels of insulin. And so we look at as well some sort of tumor markers. So we look at SOX9. SOX9 was dramatically induced and also dramatically reduced in the CHOP ASO treated mice, as was collagen 181, a marker of fibrosis. So the animals were given glucose tolerance test to further examine the metabolic effect of the CHOP knockdown. You can see there is some effect on the glucose tolerance test being slightly better. In the lower panel, the area under the curve was measured to confirm that it was specific significant effect. Excuse me. So looking further into the effects on the KC mice, we had knocked down the CHOP. Then we can see here, first of all, in the second panel compared to the first panel on the upper row, we can see the effect of the high-fat diet on the KC mice. It really promotes dramatically the advancement of the Panin lesions and emergence of early cancer in this case. And then the effect of CHOP ASO in the third panel, you can see really a slowing down of the entire process. What we like to do is quantitate these effects using an analytical software called QPath. It's freely available on the internet if you can take the time to learn it. It's very useful in analyzing all types of images. So you can see on the right-hand side, we quantitate these effects, and we can see that in the high-fat diet fed mice, they have accelerated Panin development, and that's really reduced with the ASO treatment. But even more dramatic is the effects on the acinar cells and the fibrosis. So the fibrosis was somewhat decreased, and the normal acinar cell tissue is preserved by the ASO treatment. And we measure this in the head and the tail of the pancreas. The effects are pretty similar, just a matter of degree. So that's the high fat diet effect on the pan development. That's the fatty liver that develops in these mice, and the CHOP ASO treatment abolishes the fatty liver in association with slowing down the tumorigenic effect. So finally, we get to the question of metastasis. So the mice were given either CHOP or knockout, or wild type mice were given chow diet, not high fat diet, just regular diet. And then they were injected in the spleen with KPC cells. These are, aggressive PDAC cell line. And so the cells normally will migrate to the liver, and this is a model of metastasis. So we inject the mice, and then after, I believe, 4 weeks, they were sacrificed, and we examined liver histology, metastatic tumor nodules. And remarkable effect is shown here that in the wild type mice, there are numerous liver nodules of PDAC. And really, there's a major reduction in the CHOP beta knockout livers, as you can see, 2 examples on the right. So this is a microscopic examination of wild type mouse with about 10 different sized nodules on the left, and these data are quantitated on the right. I can see I'm running out of time, but this is really the CHOP beta knockout, and you can see really no liver nodules, and again the metastasis quantitation. So this is really our working model. So of course we are studying the effects of obesogenic diet to promote the cancer, and we feel that there's an exocrine and an endocrine component to this. The endocrine component is really CHOP is permissive for high level expression of insulin. So in the presence of elevated CHOP, which is indeed caused by high level of insulin production, causes the ER stress, then you have the CHOP. CHOP is allowing the hyperinsulinemia to develop. That has a indirect or direct effects on the fatty liver development. So when you start knocking out the CHOP, you normalize the insulin, you reduce the steatosis and metastasis. And these are our acknowledgements. This is a collaboration between Cedars-Sinai and Sanford Burnham. And these are the people that did the work. Thank you very much.
Thank you. Due to time restrictions, we're going to skip the question session. You can contact Robert Richard directly or Dr. Young later for this. Thank you. Can you just skip it? Yeah, skip the questions.
Skip?
Yes, please.
So sorry.
Because you were just sorry about this. We're going to go on to the last presenter. Last presenter is Dr. Piotr Zelga from the Massachusetts General Hospital. The title of his talk is Non-Alcoholic Fatty Acid Liver Disease After Pancreatic Resections, an Underappreciated Consequence. Dr. Zelga, it's all yours.
Good morning, everyone. That's a great honor to be here and present the results of this study. We have no conflict of interest to disclose. Non-alcoholic fatty liver disease encompasses a wide spectrum of hepatic conditions, ranging from simple steatosis to steatohepatitis and cirrhosis. And its prevalence is increasing worldwide, especially in the Western population. But what's important here is just presence of NFALD is associated with a higher risk of extrahepatic comorbidity, to name cardiovascular disease, extrahepatic cancer, as mentioned by Dr. Randall, the pancreatic cancer, or even delayed recovery after anesthesia. And all these factors can modulate the clinical course and oncological treatment of patient after pancreatic surgery. So the association between the development of the NFALD and after pancreatic resection has been already studied, but there are only several papers about it, and the broad analysis, generally broader conclusion are scant. And it's due to limited number of patients which were available for that analysis, but also in a variety of radiological protocols used to assess the NFALD on the radiological imaging. In order to elucidate this important issue, we carry on this research with three main aims in mind. First of all, was to determine the prevalence of NFALD in patients after pancreatic resection. Second, to determine what risk factors are associated with the presence of the NFALD before the operation, and what factors contribute to the development of de novo NFALD. So we are talking about the NFALD after operation. For these reasons, we have analyzed 443 patients who underwent pancreatic resection at Mass General Hospital in the last decade. And in inclusion criteria were the presence of the availability of the CT scan before the operation, within 1 year, and ideally also at year 2. This patient could not develop liver metastasis, and of course, those with history of alcohol abuse and viral hepatitis were excluded from the study. The CAT scan of this patient were analyzed by 2 independent radiologists who were blinded to the medical course of these patients, and on a basis of the strict but now widely accepted protocol, were determining the presence of NFALD on a CAT scans. So if you just briefly look at the patients, majority of them underwent Whipple's operation. As you see, that diabetes were prevalent in this group. And more than half of them had the operation because of the malignant diagnosis. So they received or neoadjuvant chemotherapy, radiotherapy, or adjuvant chemotherapy. And 66% were available to analyze at the year 2. So this is the slide which presents the prevalence of NFALD in our cohort. NFALD was present preoperatively in 30% of the patients. Surprisingly, it disappeared in 2/3 of these patients by the year 1. However, 20% of the patients developed de novo NFALD. So, the total number of patients with NFALD after year 1 remain essentially unchanged. And in year 2, 35% of the patients has NFALD. So as a second objective, we find what possible risk factors are associated with the presence of an NFALD pre-op. And we have found that BMI above 25 and the presence of cancer diagnosis were those risk factors. Next, we have analyzed a subgroup of patients who developed de novo NFALD. And in this group, we have found that undergoing proximal pancreatic resections, I mean Whipple operation, receiving adjuvant chemotherapy, and presenting the signs of pancreatic enzyme exocrine insufficiency in a form of needs for enzyme supplementation and the weight loss measured here as a decrease in BMI, were independently associated with the development of new NFALD post-op. So when we analyze the subgroups of patients with persisting NFALD after year 1 with those who had de novo NFALD, we found that, yes, it's true, the importance of the extent of resection, because this patient with the new NFALD had more frequent developed newly onset diabetes mellitus and had more frequent needs for pancreatic enzyme supplementation. Conversely, when we take a broader look at the patients in which NFALD disappeared, we have found that preoperative BMI of less than 25 and age less than 65 years were associated with this favorable outcome. Moreover, we have also investigated, does the presence of NFALD preoperatively influence the rates or the severity of the complications measured according to Clavien-Dindo scale. And we have found that there's no such correlation or association. So to conclude, NFALD is present preoperatively in 30% of the patients who undergo pancreatic resection. In 2/3 of these patients, NFALD disappears after surgery, but also de novo NFALD develops in 20% of the patient. So the total numbers in our study are 31% after year 1 and 35% after year 2. De novo NFALD more frequent develops in patients who undergo pancreatectomy, receive adjuvant chemotherapy, and have pancreatic exocrine insufficiency with accompanying weight loss. Ladies and gentlemen, thanks for your attention. I welcome questions.
Thank you. Few questions from the audience. Go ahead.
Hi, Chris Wolfgang, NYU. That excellent study and really nicely presented. I was wondering if you actually looked at those with the de novo, the proportion that presented with de novo NAFLD, and looked at cancer-specific outcomes. Does it predict early recurrence or 5-year recurrence or pattern of recurrence? I was wondering if there's an interlinking, or if this is hypothesis generating that there's a link between the development of NAFL and cancer-specific features.
The female gender is in the previous studies independent risk factor also for the fatty liver. However, in these studies we have excluded those who develops a mets and so have very smooth clinical course in the terms of the cancer. So we didn't have followed the patient in that regards. Yeah, so we don't know in fact if some of them later develop liver mets or any local recurrence, and if they are alive. But we are thinking about doing that. Yeah.
But although excluded in this study, you would have that patient population to study, correct?
Yeah, so we aiming to do that. Yeah, also to say if the development of NFALD, how much it contribute to the long-term cancer-related and overall survival. Yeah, in this group of patients.
Be interesting.
Thank you. The last question.
Great talk, Somnath, University of Miami. Do you see any gender differences in your data?
Excuse me, I don't understand.
Did you observe any difference in how the females or the males responded in your studies?
No. So we here because the thing is that if we start to develop this, divide this patient into the subgroups, yeah, we just lose the statistical power. Yeah. So here, in fact, we for the purpose of getting broader results, we just keep the groups as a whole without further stratifying them to gender, for example, yeah, which is a risk factor. However, also in year 2, we are dealing with with multiple small subgroups, yeah, and this further would impair the analysis. So, well, it's still possible. We are preparing now the paper, so I will take your idea and try to look at it. Whereas now we are just, giving the big conclusions in order to see what we are standing, because in fact, it wasn't presented before. So this is the first paper which present the preoperative presence of NFALD, which is not related to the metabolic syndrome or to the typical NFALD. So this is the NFALD which is related to the pancreatic resection. And in fact, it's that the results also surprised us, and it's a food for thought. It raised more questions actually than giving us an answers.
Thank you.
Thank you. Thank you, Dr.
Well, we thank all the speakers for for tremendous presentations, really spanning a broad diversity of topics. There'll be a short break, and then we'll come back here for the endocrine pancreatitis crosstalk session. Thank you very much.